Rice currently, we have about 20,000 cultivars connected from 116 countries, almost every country where rice is grown. And once we got them, we get them characterized, grow them out, and conduct all kinds of research to get them, to get all the characteristics, and so that way, when breeders trying to select the parents, they will data will help them to get what they want. Now, for example, so that, you say that that rice there have a different height and some taller, some shorter, and with the pipe. That's the example for our characterization. That's a characterization for what? For the force mark. So we have that's a we from a 20,000 cultivar we sampled 10%. Just randomly sample with certain rules. Make sure they have a good representation of the 20,000, and then we further select 10% out of that. So that means 1%. Because that's a force mark is, it's very hard to evaluate. So, but we make sure this number is good enough to handle. But also they have to represent our entire variation. That's what it did. Come out 200 lines there. And we conduct a replicator test and for the force mark. That's just one example. We have 27 characteristics to characterize our 20,000 cultivars. Once we have them, we have all the cultivars characterized, and also we extract their DNA and conduct the research of describe their genetic relationship. So we get the information which variety genetically far from another, which variety is closer to another. So that way, give to this information help breeders to design their cross strategy, choose the right parents, and also we develop a genetic marker, just like that. What do? We have use a rice entire genome. We use wide coverage genetic markers for the entire genome. And then with this side, we have a phenotypic variation, just like a force mark. And then we use a statistical package software to see which marker on which location in the chromosome that co-variate with the phenotypic trait. Then that marker is associated to the phenotypic trait. Then that marker can be used for the marker system breeding. So that's what we have genetic structure and the developed marker for each important trait. Currently, we have developed marker for blast and cooking quantity, just like amylose content, and also for the yield potential, and for the stigma exertion, which is important for hybrid seed production. I guess we have a bunch of the guests from Rice Tech, which is work on the hybrid rice. And by the way, we have an important guest here today. Is Dr. Chen Xihua, from Director General from China National Rice Research Institute. Welcome. So this is what we do, and also with this information to public available, and also the germplasm, everywhere germplasm is public available. That's what also we have on the germplasm research. We develop cultivar also. Here's some example of the cultivar developed in our program. This program. We do not like Dr. Morton or Dr. Gibbons spend our major job is not breeding, but we do breeding. If we say some things can easily to be done, like this one, some Rondo, Jazz, Delta Bell, and Clustering Gold. Those just our germplasm characterization, we saw some of them very promising, but just have a few traits need to improve. Just like a roundel, it's a line of germplasm introduced from China, was too tall, and also the amylose, the cooking quantity is not fit our market. So we just conduct the mutation breeding to improve those traits and become a cultivar, which released last year. It's good for the typical long grain, high quality, and the parboil, good for parboil industry business. And also the classic Delta Bell is the aromatic rice, and also the Jess is a typical aromatic rice introduced from Thailand, and which was originally was too tall, with very easy to lodge, and also the flower too late. So we just conduct the mutation breeding, use the gamma ray to shoot the seeds and make a change, and then from offspring we find that's type of change is exactly what we're looking for. So we got this cultivar and this release there. We have a big field, demonstration field down this way, and if anybody interested, we can arrange a special tour to the actual field.